Osteoarthritis is a condition that impacts hundreds of millions of people, and for the most part, treatments have focused on managing symptoms, not finding a cure. But what if the key to protecting our joints is already inside our own cells? Today, we're going to examine new research that's uncovered what you could call a molecular shield, a single protein that seems to guard cartilage against the damage of aging. We'll explore how it works and whether it could point toward the future of treating this very widespread condition.
So, to really get a handle on this discovery, we're going to break it down into 5 key parts. First, we'll establish the scale of the problem with aging joints. Then, we'll trace the search for this protective protein and look at how its molecular shield actually functions. After that, we'll see if it can reverse damage in a preclinical model. And finally, we'll explore how modern mRNA technology might one day be used to deliver this therapy.
So let's start with context. Why is this line of research so important? Well, it begins with a problem that many of us are familiar with, either directly or indirectly: osteoarthritis. It's one of the most common and often debilitating disorders associated with the aging process.
And the scale of this is significant. Right now, nearly 600 million people around the world are living with osteoarthritis. It's a condition that goes beyond just causing pain. It can severely limit mobility and, as a result, impact many different aspects of a person's life.
So what's actually happening on a physical level inside the joint? On the left here, you can see a healthy joint, and the key feature is that smooth, intact cartilage that allows for easy movement. But on the right, in an osteoarthritic joint, that protective cartilage is breaking down. At the same time, you can get the formation of painful bone spurs and inflammation in the lining of the joint. All of these factors contribute to the pain, stiffness, and loss of motion that define the disease.
We know that factors like aging and mechanical stress contribute to osteoarthritis, but the precise molecular triggers that set it off have been harder to pin down. This particular research set out to solve that mystery by searching for a protective mechanism hidden within the very cells that are responsible for building cartilage, the chondrocytes.
A key process in this joint degradation is something called chondrocyte senescence. Now, that might sound complex, but you can think of it simply as the cartilage-building cells getting old and stopping their normal function. They can no longer properly repair and maintain the cartilage, and as they decline, the entire joint structure begins to degrade with them.
So how did they find the specific protein involved? Well, the researchers started with 23 potential candidates, a family of enzymes called palmitoyltransferases. In human cartilage cells, they used a methodical approach, turning off the gene for each one of these enzymes, one by one, to see what would happen. And when they turned off the gene for one specific enzyme, ZDHHC11, they saw a clear effect. The cells started showing signs of aging and cartilage breakdown. They had found their key protective factor.
And this wasn't just an observation they made in a lab dish. When the researchers analyzed actual cartilage samples from aged mice and from human patients with osteoarthritis, they found a very distinct pattern. The healthier parts of the cartilage had plenty of ZDHHC11, but the degenerated, damaged areas had significantly lower levels. This provided strong evidence that a loss of this specific protein is a key feature of the disease as it progresses.
So, having identified ZDHC11 as a key protector, the next logical question was, how does it actually work? This next part of the study details the specific biochemical mechanism through which ZDHC11 shields these cartilage cells.
The process that ZDHC11 uses is called S-palmitoylation. It's a chemical modification where the enzyme attaches a small fatty acid, a kind of lipid tag, onto another protein. This seemingly small addition acts almost like a molecular switch, capable of changing how that target protein behaves inside the cell. And this protective pathway really boils down to 3 main components. First, of course, is our enzyme, ZDHC11. The second is the protein that it protects, which is called APOD. And the third is a protein named GNB2, which is part of the cell's natural machinery for marking other proteins for disposal. The mechanism itself unfolds in a clear sequence of events. First, ZDHC11 attaches that palmitoylation group to the APOD protein, creating a sort of physical shield. The purpose of this shield is to block GNB2 from being able to bind to APOD. GNB2's normal job is to tag APOD for destruction, so by preventing this interaction, ZDHC11 stabilizes APOD and protects it from being degraded. And it's the presence of this stable APOD that, in turn, helps maintain the health of the cartilage cell.
And to put it in the precise language of the study itself, the researchers concluded that ZDHHC11-mediated palmitoylation inhibits the ubiquitination and degradation of APOD. In functional terms, that ubiquitination is the destroy me tag, so the protective shield attached by ZDHHC11 physically prevents that tag from being added, effectively saving a beneficial protein from the cell's own disposal machinery.
Demonstrating how this works in a cell culture is a critical first step, but the real test is whether it can have a therapeutic effect in a living organism. To find that out, the researchers moved on to a preclinical mouse model of osteoarthritis.
The experiment was conducted on aged mice, in which osteoarthritis was surgically induced by destabilizing the medial meniscus. This is a standard model for studying the disease. After the injury, the mice received a single injection directly into the joint. This injection contained a harmless virus, an AAV, which was engineered to deliver the gene for ZDHC11, thereby increasing its production. The outcomes were then evaluated 8 weeks later.
So, the first key result was a reduction in the formation of bone spurs. On this chart, you can see the middle bar, labeled DMM, shows a high number of these osteophytes in the mice with untreated osteoarthritis. But if you look at the bar on the right, that's the group that received the ZDHC11 therapy. The number of osteophytes is significantly lower. This indicates a clear protective effect against this particular feature of the disease.
Next, they examined the cartilage damage itself. This was measured using a standard grading score, where a higher number means worse damage. The untreated DMM group there in the middle shows a high score, indicating severe cartilage loss. But again, the group treated with ZDHHC11 had a much lower score, which confirms that the therapy was protecting the cartilage tissue from degradation.
In summary, these preclinical animal studies provided an important validation. Supplementing ZDHHC11 directly within the affected joint was effective at mitigating key pathological signs of age-related osteoarthritis in this particular mouse model.
The viral gene therapy was an effective proof of concept, but the researchers also tested a more modern and potentially more precise approach. This final part of the study explores a delivery system that uses messenger RNA, or mRNA, to specifically target the damaged cartilage cells. The idea here is a subtle but important shift from permanent gene delivery to providing a temporary instruction. So instead of using a virus to install a new gene, this approach delivers mRNA. You can think of mRNA as a temporary blueprint. It's delivered to the cartilage cells, which then use it to produce the protective ZDHHC11 protein themselves for a limited period of time, after which the blueprint naturally degrades. Now, to achieve this, the researchers designed a multi-part delivery system. The mRNA payload is encapsulated inside a lipid nanoparticle, an LNP, which protects it on its journey. And critically, the surface of this nanoparticle is decorated with something called a chondrocyte affinity peptide. This peptide acts as a homing signal, ensuring the nanoparticle binds specifically to cartilage cells, which helps minimize off-target effects.
And when this targeted mRNA system was tested in a rat model of osteoarthritis, the results were quite comprehensive. As this table summarizes, the treated group showed significant reductions across every major sign of the disease: fewer bone spurs, less cartilage damage, and also reduced joint inflammation. This suggests the targeted delivery was effective across multiple metrics of the condition.
So, from discovering a single protective protein to delivering its instructions with targeted mRNA, this research really opens up a new potential strategy. And it leaves us with a compelling question for the future. Could temporarily reprogramming our own cells in this way be a key to treating not just osteoarthritis, but perhaps many other complex diseases of aging?